Each bacterium contains a specific set of enzymes that allows it to perform unique biochemical functions. This variation is the basis for identifying bacteria through biochemical tests, as distinct enzymatic reactions lead to observable changes. These differences can be exploited to classify and distinguish various bacterial species in a laboratory setting effectively. For instance, E. coli and Klebsiella pneumoniae are both gram-negative bacilli, and they grow as pink colonies on MacConkey agar due to their ability to ferment lactose. However, despite this similarity in appearance, they differ markedly in their biochemical reactions. This distinction allows microbiologists to accurately identify these two organisms during laboratory testing. The key to differentiating these two bacteria lies in their biochemical reactions. For example, E. coli tests positive for indole production while testing negative for citrate utilization. In contrast, K. pneumoniae tests negative for indole and positive for citrate. These specific reactions highlight how biochemical tests serve as a powerful tool for accurate bacterial identification. Biochemical tests can be categorized based on the metabolic processes and specific enzymes they target. This classification is crucial as it facilitates the systematic identification process for various bacteria. Understanding the metabolic pathways is essential for microbiologists to select the appropriate tests during bacterial identification. The identification process begins with a clinical specimen, which is cultured to obtain a pure colony. This colony undergoes a gram stain for preliminary identification. Once an initial assessment is made, microbiologists select appropriate biochemical tests. The results from these tests provide critical data that lead to the final identification of the bacterial species. Each step is logical and sequential, building upon the last for accurate identification. In the process of aerobic respiration, bacterial cells produce harmful byproducts known as reactive oxygen species, one of which is hydrogen peroxide, or hydrogen oxide. These byproducts can be damaging to the bacteria themselves, affecting their DNA, proteins, and cell membranes. The catalase test serves a specific purpose. It determines whether a bacterium produces the enzyme catalase. This enzyme plays a crucial role in protecting bacteria from the harmful effects of hydrogen peroxide, thereby aiding in their survival during aerobic respiration. When conducting the catalase test, an immediate formation of bubbles indicates that the bacterial culture is catalase positive. The presence of bubbles signifies that catalase is breaking down hydrogen peroxide into water and oxygen, effectively demonstrating the bacterium's enzymatic ability. Conversely, if no bubbles develop during the catalase test, it suggests that the bacterium does not produce catalase. This outcome classifies the organism as catalase negative, which provides valuable information in the identification process. The oxidase test is particularly useful in distinguishing between various types of gram-negative rods. This differentiation helps in identifying specific bacterial species within this broad category, allowing for accurate microbiological assessments. Bacteria that test positive for oxidase possess a specialized enzyme known as cytochrome oxidase. This enzyme enables the bacteria to effectively utilize oxygen during respiration, which is a vital metabolic process for many organisms. In the presence of cytochrome oxidase, a colorless reagent used in the oxidase test quickly changes color to deep purple. This color change confirms the enzyme's presence, thereby providing insight into the bacteria's identity. The coagulase test is specifically designed to differentiate between species within the Staphylococcus genus, focusing on identifying pathogenic strains like Staphylococcus aureus. This identification is crucial in clinical settings where specific treatments may be required. Coagulase acts as a blood clotting enzyme, a feature that allows pathogenic bacteria to form protective barriers. This ability enhances their survival and pathogenicity, making it a significant factor in bacterial identification. When Staphylococcus aureus is mixed with liquid blood plasma, the coagulase enzyme converts fibrinogen, a soluble protein, into solid fibrin threads. This reaction leads to the formation of a visible clot, indicating the presence of coagulase in the bacterial sample. The clotting of the liquid plasma results in a thick, gelatinous mass, which serves as a visual confirmation of the coagulase test. This positive reaction is critical for differentiating pathogenic staphylococci from non-pathogenic varieties. The indole test relies on the presence of the enzyme tryptophanase, which some bacteria possess. This enzyme breaks down the amino acid tryptophan, resulting in a byproduct known as indole, which has a distinct odor. This biochemical reaction is significant for specific bacterial identification. To determine whether indole has been produced, Kovach's reagent is introduced to the testing sample. This reagent acts as a chemical detector, reacting with indole to produce a visual color change. In cases where the bacteria lack tryptophanase, tryptophan remains undigested, and no indole is formed. In such instances, Kovach's reagent retains its yellow color, which indicates a negative reaction, crucial for distinguishing bacteria like E. coli from Klebsiella pneumoniae. The indole test is particularly important for identifying gram-negative intestinal bacteria. Specifically, it helps differentiate between E. coli, which tests positive for indole, and Klebsiella pneumoniae, which tests negative. This distinction is vital for accurate diagnosis in clinical microbiology. The urease test is particularly effective for identifying rapid urea splitters such as Proteus mirabilis. This bacterium is commonly associated with urinary tract infections and can be distinguished from other gram-negative rods that do not split urea. This distinguishing feature is essential for appropriate treatment decisions. Certain bacteria possess an enzyme known as urease, which breaks down urea. The breakdown of urea typically releases ammonia gas, which can significantly alter the acidity of the environment. Understanding this biochemical reaction is key to utilizing the urease test for identification. The urease test employs an agar medium containing a color-changing indicator called phenol red. When the environment becomes alkaline due to ammonia production, phenol red changes from yellow to vibrant pink. This color shift indicates positive urease activity and aids in bacterial identification. The methyl red (MR) test assesses whether an organism produces highly stable organic acid end products through glucose fermentation. This capability is significant for understanding the metabolic processes of different bacterial species. Mixed acid fermenters, when exposed to glucose, convert it into stable organic acids. These acids include lactic acid, acetic acid, and succinic acid, permanently lowering the pH to 4.4 or lower. This characteristic is crucial in differentiating fermentative bacteria during testing. Upon adding the methyl red indicator, a vibrant red color indicates high acidity, confirming the presence of stable organic acid production. This reaction provides significant insight into the fermentation capabilities of the tested organisms. Negative organisms produce neutral end products that maintain a pH above 6.2, resulting in the agar retaining its yellow color. This outcome will help clarify which bacteria exhibit specific fermentation characteristics during analysis.